Hello everyone, myself Manju Sharma from Suresh Kanviar University, Jagatpura, Jaipur, and the department is School of Agriculture, and the subject is plant pathology. So today we are going to discuss about plant disease epidemiology. So let's come to the what is plant disease epidemiology. So here two terms which we are going to discuss like epiphytology or epidemiology. So a study of the rate of multiplication of a pathogen and spread of the disease caused by it is in a population. Epidemiology deals with outbreaks and spread of diseases in a population. Importance of epidemiology. Knowledge of epidemiology is useful in forecasting of a disease and also for the management of a disease. So here the terms compound interest disease and the simple interest diseases were given by Vander Planck in 1963 in his book that is Plant Disease Epidemics and Control. So, what is the compound interest disease and what is simple interest disease? And we can say polycyclic or monocyclic. So, rate of decrease of disease is mathematically analogous to compound interest in money, like interest is added periodically to the capital and interest gets interest. That is polycyclic or compound interest disease. Another is the simple interest disease or monocyclic. Rate of increase of disease is mathematically analogous to simple interest in money. Interest is added only at the end and interest does not get interest. Then the pathogen produce spores at rapid rate and the pathogen produce spores at very slow rate. So the propagules disseminated by air in polycyclic disease and in monocyclic disease the propagules disseminate by soil or seed. In polycyclic disease, the incubation period and sporulation period is short, or in monocyclic disease, the incubation period and sporulation period is long. In polycyclic, there are several generations of the pathogen in the life of a crop, and in monocyclic, there is only one generation of the pathogen in the life of a crop. For example, in polycyclic, like rust of cereals, and in monocyclic, smut of wheat, barley, and sorghum. After that, disease triangle and disease pyramid, that is tetrahedron. So, first of all, what is disease quadrangle? Four fundamental elements that are required for a disease in a plant are a susceptible host, a pathogen capable of causing disease, favorable environment, and adequate time. So, first is what is important for a biotic disease to occur. Like, first of all, environmental condition should be in favor of the pathogen, like moisture and temperature. Secondly, environmental factors encourage or discourage the susceptibility of the host and the pathogenicity of the pathogen. Then, the environmental diseases are caused by persistent, unfavorable environmental conditions. So let's look at these factors individually. Like first is the host factors. So distance of susceptible plants from the source of primary inoculum, abundance and distribution of susceptible host, continuous cultivation of a susceptible variety over a large area, like buildup of inoculum and decrease the chances of epidemic disease provenance in the host due to environment, host nutrition, excessive application of nitrogenous fertilizer, etc. So there is the presence of suitable alternate or collateral host. Next one is the pathogen factors. Pathogen factors like presence of virulent aggressive isolates of a pathogen. There is a rapid cycle of infection is essential, high birth rate, enormous quantity of spores that are adapted to long distance dissemination in a short time. Then low death rate of the pathogen and easy and rapid dispersal of the pathogen and ultimately adaptability of the pathogen. So these all are the pathogen factors. Next factors are weather factors in which like optimum weather conditions for sporulation, dispersal, infection and survival of pathogen. These conditions includes so the condition includes like optimum temperature, moisture, light, etc. for the development of an epidemics. Science which deals with the relationship between weather and epiphytotic that is called meteoropathology. The next thing is the effect of time on disease development become apparent when we consider so importance of time of year, then duration of frequency of favorable temperature and rain duration the cycle of a particular disease. So here four things are there like environment, pathogen, plant, and time, and they all are commonly called the epidemic, disease epidemic. So next one is the disease forecasting. The aim of the disease forecasting is to control disease, to suppress pathogen from altering one or more sides of the disease triangle. This require knowledge about the disease forecasting, greater knowledge of the disease situation than is available in most cases. Then the forecasting is predicting for the occurrence of plant disease in a specified area ahead of time. Why it is done? If we can predict the probable outbreaks or increase in intensity of disease, we can be well prepared. So take suitable control measures in an advance to control or to avoid the losses. So it involves like well organized teamwork, expenditure of time, energy and money, then uses of forecasting. First is it is used as an aid to the timely application of chemicals control measures. Then secondly is the example of first spray warning services like grapevine, downy mildew, forecasting scheme in example of first spray warning services and the grapevine downy mildew forecasting scheme in France, Germany and Italy in 1920. Disease forecasting method are available for the flowering plant diseases. So first is the plant diseases and countries. Grapevine downy mildew disease in Australia, France, Germany, Greece, Italy, Romania, Spain, USSR and Yugoslavia. Then cucurbit downy mildew in USA, potato late blight, Australia, Brazil, Finland, France, Germany, Greece, Japan, Netherlands, Norway, Peru, UK, and the USSR. Tobacco blue mold disease in Canada and USA. Apple and pear scab disease in Australia, Canada, Netherlands, New Zealand, sugar beet root rot in USA, wheat bone rust in USA, then wheat brown leaf rust in USA, and the corn bacterial wilt in USA, and the sugar beet curly top is in USA. So, what all information is needed for disease forecasting? Forecasting diseases is a part of applied forecasting disease is a part of applied epidemiology. Hence, the knowledge of epidemiology, that is the development of disease under the influence of factors associated with the host pathogen, is necessary for accurate forecasting. Hence, the knowledge of epidemiology, that is development of disease under the influence of factor associated with the host pathogen and is necessary for the associated with the host pathogen is necessary for accurate forecasting. So the factors of so it is necessary for accurate forecasting. So the factors of epidemic or abiotic. So the factors of epidemic and its components should be known in advance before forecasting is done. Then the factors are in which the first factor is the host factors. So there is the prevalence of susceptible varieties in the given locality, response of host at different stages of the host, prevalence of susceptible varieties in the given locality, response of host at different stages of the growth to the activity of pathogen. For example, some diseases are found during seedling stage, some diseases are found during seedling stages while other attack then the density and distribution of the host in a given locality in which basically dense population of susceptible variety invite the quick spread. So dense population of susceptible variety which invite quick spread of as epidemic, growing susceptible varieties in scattered location and there too in a limited area where they less prone to epiphytotic. Then the second factor is the pathogen factors. Amount of primary initial inoculum in the air, soil or planting material, dispersal of inoculum, spore germination, incubation period, sporulation on the infected host, redispersal and dissemination of spores, then pre-nating stages like live throughout a number of years, inoculum, pre-nating stages like live throughout a number of years, and inoculum potential and density in the seed, soil and air. Now, the third thing is the information about environmental factors. So, in which, like temperature, humidity, light intensity, and wind alkalinity, and the wind velocity, light intensity, humidity, and temperature. So now requirement or conditions for disease forecasting. So the disease must cause economically significant damage in terms of yield, loss or quality. Control measures must be available at an economically accepted mean or acceptable cost. So the disease must vary each season in the timing of the first infections and its subsequent rate of progress. So the disease must vary each season in the timing of the first infection and its subsequent rate of progress. If it is does not, there is no need for forecasting. So the growers must have sufficient manpower and equipment to apply control measures when disease warming is given. Then the growers must have sufficient manpower and equipment to apply control measures when disease warning is given. Long term warning or production are most useful than short term warning of production. Now, the criteria or model used in making a prediction. So, first thing must be based on sound investigational work that is carried out in the laboratory, must be based on sound investigational work carried out in the laboratory and in the field and tested over a number of years. And this is done to establish its accuracy and applicability in all the locations where it's used in investi- is investigate. Then the methods of disease forecasting and how it is done. So first point, like forecasting based on primary inoculum, forecasting based on weather conditions, forecasting based on correlative information, and the fi- and the use of computer for disease forecasting. Then first point, forecasting based on primary inoculum, like determination of presence, density, and viability of primary inoculum determined in the air, soil, or planting material. Assessing occurrence of viable spores or so there is the assessing occurrence of viable spores or propagules in air trapping device that is called spore traps. Next one is soil-borne disease. Soil-borne disease, primary inoculum, that is determination by monoculture method. Then seed testing can be done and accordingly chemical seed treatment Then seed testing can be done and accordingly chemical seed treatment. Then different seed testing methods like presence of loose smut of wheat, ergot of pearl millet, and there is the different seed testing method in which like presence of loose smut of wheat, ergot of pearl millet, and viral diseases of So there is a presence of loose smut of wheat, ergot of pearl millet, and viral diseases of potato can be detected in seed pods. And the extent of many virus diseases is dependent on the severity of the preceding winter, which affects the size of vector population in the growing season. So that is the disease forecasting based on weather conditions. Weather conditions like temperature, relative humidity, rainfall, light, wind velocity. So during the crop season and during the intercrop season are measured. So the weather conditions above the crop and at the soil surface and season are measured, then weather condition above the crop and at the soil surface are also recorded. Then the forecasting based on correlative information, like first is the weather data of several years are collected and correlated with the intensity of the disease. So the data are compared and then the forecasting of the disease is done. Forecasting criteria develop from comparison of disease observation with standard meteorological data have been provided for diseases like septoria, leaf blotch of wheat, fire blight of apple and barley, and powdery mildew. So, forecasting criteria develop from comparisons of disease observation with standard meteorological data have been provided for diseases like septoria, leaf blotch of wheat, fire blight of apple and barley, powdery mildew. Then the use of computer for disease forecasting. In some advanced countries, forecasting of disease is made by the use of computers. This system gives the result quickly One such computer based on program in the USA is known as Ble- Blindcast for potato lead blight. Examples of other well-developed forecasting system like early and late leaf spot of groundnut, blister blight of tea, southern corn leaf blight, rice blast, wheat stem rust, and brown striped downy mildew of corn. After that, remote sensing. So basically, the remote sensing is estimating an object phenomenon without being in physical contact with it. Remote sensing is a science art that permits us to obtain the information about an object, a phenomena through analysis of data obtained through sensory devices without being in physical contact with that object. So, objectives of remote sensing in plant pathology. Here, the assessment of disease over a vast area to know the relationship of diseases and environment, to know the origin and development of epidemics, and the quantitative assessment of the diseases. Then, remote sensing techniques. Here, like two techniques are there, aerial photography and the another one is the satellite remote sensing. First is aerial photography. Then Colwell in 1956 first used remote sensing technique for monitoring same rust of wheat. The key to distinguish diseased and healthy part of a crop is to use appropriate film or filter combinations. The main film types used are panchromatic, infrared, normal color, and color infrared. The infrared films are preferred because of their superior sensitivity to visible light and to near infrared wavelengths of radiation 700 to 900 μm. The color infrared of Ektachrome arrow infrared, that is camouflage detection film, is superior as it can show the difference between diseased and healthy patches of plant in color. Then the aerial photography. Aerial photography. How does NIR works? Leaves have chlorophyll and spongy cell structure. Chlorophyll absorb red, that is 600 to 700 nanometer, and blue 400 to 500 nanometer light. Chlorophyll reflects green light, it is 500 to 600 nanometer light. Spongy cell structure which reflects near infrared light, that is 700 nanometer, and absorb Less chlorophyll and less spongy cell means that is unhealthy tissue. Unhealthy vegetation has less chlorophyll and less spongy cell structure. Less spongy cell structure means less reflection and more absorption of near infrared light. So therefore reflects less NIR light. So here like unhealthy vegetation has less chlorophyll and less spongy cell structure. So less spongy cell structure means less reflection and more absorption of IR light and therefore reflects less NIR light. So here also unhealthy vegetation has less chlorophyll and less spongy of cell structure, and in another one the less spongy cell structure means less reflection and more absorption of light. So here these are images of cotton field infested with cotton root rot from approximately 1927. So they were all taken at angle except so distances vary depending on the location. In the image due to perspective image was taken from directly overloaded, so in one scale is equal throughout the image, making too easy to determine how large this dead patch was. Up to here, remote sensing and their importance. Thank you. Gene cloning, recombinant DNA technology, and genetic engineering. Steps in gene cloning, identification and isolation of desired gene or gene fragment to be cloned, restriction digestion and electrophoresis. Insertion of the isolation gene in a suitable vector, that is ligation. Introduction of this vector into a suitable organism or cell called host, that is transformation. Selection of transformed host cells, selectable markers, multiplication, integration, followed by expression of the introduced gene in the host. Enzyme involved restriction endonuclease, DNA ligase, DNA polymerase, RNA polymerase, and reverse transcriptase. Vectors used in gene cloning. A vector is a DNA molecule that has the adaptability to replicate in an appropriate host cell and into which the DNA fragment to be cloned, that is called insert. DNA insert is integrated for cloning. Example, tumor inducing TI plasmid of Agrobacterium tumefaciens PBR322, bacteriophages, cosmid vectors, and derived from phase lambda. The process of gene cloning is used to locate and copy a specific gene from all the DNA in an organism. TA plasmid of Agrobacterium tumefaciens vector. TA plasmid is a large consecutive plasmid or megaplasmid of about 200 kb. Conjugative plasmids contain a set of transfer or tragenes which promote sexual conjugation between different cells, may be transferred from one bacterium to another via sex pili. TA plasmid has a tumor tDNA region 15 to 24 kb, which is bounded by a pair of 24 base pair repeats, carries genes for auxin, cytokines, and opine synthesis, which are responsible for tumor formation, and that is tumorigenesis. The transfer of tDNA depends on 35 kb virulence virgin region of Ti plasmid, 7 operon ranging from vir A to vir H, vir A, vir B, then vir C, vir D, vir E, vir G, and vir H. Operon is a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. The protein products of these genes respond to phenolics to generate a copy of tDNA. So, transfer of tDNA depends on 35 kb virulence where region of the Ti plasmid, then there will be 7 operon. Operon is a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. The protein products of these genes respond the phenolics to generate a copy of. Operon is a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. The protein products of these genes respond to phenolics to generate a copy of tDNA and mediated its transfer into. So the protein products of these genes respond to phenolics to generate a copy of tDNA and mediates its transfer into the cell. The tDNA, when transferred from the agrobacterium to the plant cell, integrates with the chromosome, and the plant cells which are affected begin to synthesize opines, auxins, and cytokines. So here that is the Ti plasmid. First is the origin of replication, then transfer of tDNA. So basically transfer of tDNA which depends on 35 kg virulence or vir region. So here auxin biosynthesis 24 repeat left border cytokines biosynthesis, opine synthesis, 24 base pair repeat, right, Buddha? Now, the opines are tumor-specific compounds formed by the condensation of amino acid, virgin, keto acid, and sugar. The this opine, the opines that is octopine, nopaline, succinimopine, or leucinopine can be metabolized only by agrobacteria. So the IAA, oxin and isopentyl, that is cytokine, are phytohormones which cause the proliferation of plant cell and fruits. And there is the induction of the gall. Then plant-bound exudates contain phenolates which attract agrobacterium. The strong virgin induce an syringic acid, fertile fulic acid, acetylcyanogen, and snapping acid, and slow and only agrobacterium with Ti plasmid are attracted by these compounds. Thank you.